Thank you. Good evening. I'm John Downing. I'm chair of the executive board of the Council of Scientific Society Presidents. And on behalf of CSSP, it's my pleasure to welcome you to the Fred Kavli Science at the Frontiers lecture, featuring Dr. J. Craig Venter. The Council of Scientific Society Presidents is dedicated to advancing leadership in science and technology. We do this in a variety of ways, but through our meetings, advocacy, and programs aimed at public understanding of science. Tonight's Kavli lecture is an example of our goal to improve the public understanding of science. I'm especially delighted to have here tonight Dr. Myung Chung, who is out here. Could you wave? And she's Executive Vice President of Science Programs of the Kavli Foundation, which is dedicated to advancing science for the benefit of humanity, promoting public understanding of scientific research, and supporting scientists in their work. Thank you so much, Dr. Chun, for being here. We appreciate it so much. And also, please accept our sincere appreciation for the Kavli Foundation for its support of these lectures and also the live streaming video that's going out. We appreciate it. And now it's my privilege to introduce Dr. J. Craig Venter. J. Craig Venter, PhD, is regarded as one of the leading scientists of the 21st century for his numerous invaluable contributions to genomic research. He is co-founder, executive chairman, and CEO of Human Longevity Inc., or HLI, a San Diego-based genomics-based technology-driven company creating the world's largest and most comprehensive database of whole genome, phenotype, and clinical data. HLI's goal is to extend healthy human life, and I certainly appreciate that, by revolutionizing human health and transforming the practice of medicine. In addition to his role at HLI, Dr. Venter is founder, chairman, and CEO of J. Craig Venter Institute, or JCVI, a not-for-profit research organization with approximately 200 scientists and staff dedicated to human, microbial, plant, synthetic, and environmental genomic research and the exploration of social and ethical issues in genomics. Dr. Venter is also co-founder, executive chairman, and co-chief scientist of Synthetic Genomics Inc., or SGI, which is a privately held company focused on developing products and solutions using synthetic genomics technologies. Dr. Venter began his formal education after a tour of duty as a Navy corpsman in Vietnam from 1967 to 1968. After earning both a bachelor's degree in biochemistry and a PhD in physiology and pharmacology from the University of California at San Diego, he was appointed professor at the State University of New York at Buffalo and the Roswell Park Cancer Institute. In 1984, he moved to the National Institutes of Health campus, where he developed expressed sequence tags, or ESTs, a revolutionary new strategy for rapid gene discovery. In 1992, Dr. Venter founded the Institute for Genomic Research, TIGR, now part of the J. Craig Venter Institute, a not-for-profit research institute, where in 1995, he and his team decoded the genome of the first free-living organism, the bacterium Haemophilus influenzae, using his new whole genome shotgun technique. In 1998, Dr. Venter founded Celera Genomics to sequence the human genome using new tools and techniques he and his team developed. This research culminated with the February 2001 publication of the human genome in the journal Science. He and his team at Celera also sequenced the fruit fly, mouse, and rat genomes. Dr. Venter and his team at JCVI continue to blaze new trails in genomics. They have sequenced and analyzed hundreds of genomes and have published numerous important papers covering such areas as environmental genomics, the first complete diploid human genome, and the groundbreaking advance in creating the first self-replicating bacteria cell constructed entirely with synthetic DNA. Dr. Venter is one of the most frequently cited scientists and the author of more than 280 research articles. He is also the recipient of numerous honorary degrees, public honors, scientific awards, including the 2008 United States National Medal of Science, the 2002 Gardner Foundation International Award, the 2001 Paul Ehrlich and Ludwig Darmstädter Prize, and the King Faisal International Award for Science. Dr. Winter is a member of numerous prestigious scientific organizations also, including the National Academy of Science, the American Academy of Arts and Sciences, and the American Society of Microbiology. What a pleasure it is to have you here. Tonight, Dr. Winter will talk about a subject that's near and dear to every one of us, of course, human longevity. Dr. Winter, please.
Well, thank you very much for the, that's not a beer can, it's the warm introduction, and it's a pleasure to be here, and so thank you to the Council of Scientific Presidents and the Kavli Foundation for hosting me here. So I'm going to walk you through a lot of things going on at the leading and bleeding edge of genomics. Some of this is taking place in this building, the Human Longevity building in La Jolla, California. This building here is the Venter Institute in La Jolla. It's on the UCSD campus. It's the nicest spot because it overlooks La Jolla Cove. And so it's a wonderful environment. But speaking of the environment, it's the first carbon-neutral research building on the planet. It generates all its own power and does lots of recycling. So a lot of unique engineering went into this. And there's carbon-neutral office buildings, but trying to get a full-blown research institute with the power requirements needed at a carbon-neutral was a huge challenge. And the architects, ZGF, in Los Angeles have won now numerous awards for their design in this building. So in all these different organizations, we're focused on trying to infer or interpret your phenotype from the genome sequence. And the question has been for a long time whether these things were predictable. We've started early on reading the genetic code, and I call this digitizing biology, where we take the 4-letter chemical code of ACs, Gs, and Ts, and when we sequence those, we convert them into the ones and zeros in the computer. And this has been going on at ever-increasing paces, starting in the late 70s, but it really took off, as you heard, when my team sequenced the first genome in 1995. So we had the 20th anniversary of that this June of our paper in Science. This was new techniques that allowed us to use computer tools to assemble things to go a lot faster. And it was only 5 years after this first bacteria that we were able to scale up to do the first human genome sequence. That took our team 9 months and cost $100 million, but that's in contrast to a multi-billion-dollar federal program, and including over $2 billion from other countries thrown in that was supposed to take 15 years. So the change in pace of this field is started from the outset and is going even faster. Now, we're also learning to write the genetic code. So there's lots of things we want to do, starting with the ones and zeros in the computer. We want to be able to interpret them, see what they say about all of us. But part of the goal in doing that is to see if we could rewrite the genetic code, create chemicals from scratch, driven just by what was in the computer and nothing else. So we started with a small virus, FIX-174. It took a year or so to learn how to do this, but we wrote its genome, injected the DNA into E. coli. E. coli read that DNA as though it was normal DNA, made the proteins that were coded. The proteins self-assembled, made the virus. The virus made more copies of itself until it killed the cells, and then went on to infect other cells. So this is a sketch of what the virus looks like. But we call this, for the first case, where the software is building its own hardware. We just put in a piece of chemical software and end up with a virus. And now, if we could do that with computers, things would move a whole lot faster. Our goal was to build an entire bacterial genome, not just a virus that hitchhikes on our genomes. And it took lots of different technical tools over almost a decade period of time. All this work was funded by the company Synthetic Genomics, and it was done almost entirely at the Venter Institute. Ham Smith, the Nobel laureate, was one of the team leaders. Dan Gibson developed a whole new technique for rapidly putting pieces of DNA together that we could assemble. That's now called the Gibson assembly. And we finally were able to design and build this 1.1 million base pair genome by taking 1,000 base pair pieces, putting 10 of those together to make 10,000 base pair pieces. We put 10 of those together to make 100,000 letter pieces. And we put 11 of these pieces into yeast, and yeast assembled them into the genome for us. So we mastered the chemistry. It was long and hard slog. And now Synthetic Genomics actually has robots that does this. The next stage was a little bit more difficult, learning how to transplant a genome. And so we have this very high-tech movie to show you what we think happened. So we injected the synthetic DNA into the cell, into this recipient cell. And now we have a case where there's one cellular body, one phenotype, but it has two sets of genetic instructions. And just like Phi-X, this new piece of DNA started to be read, and proteins started to be made. Some of the early proteins were restriction enzymes that recognized the chromosome in the cell as foreign DNA and chewed it up. So now we have just the body of one species and the genetic instructions of another. And what happened in a very short period of time is this got converted into what was totally driven by the new genetic instructions. In these cells, there's not a single molecule of the original cell that was used as the recipient left. Every single molecule in them derived from the instructions from the genome that we transplanted. So this was 5 years ago in July of 2010 where this was reported. We wanted to go a whole lot further, a lot faster. We've gone further. Faster was not going to happen. We wanted to design from de novo design in the computer a minimal cell. And we thought we learned enough from all these different studies that we should be able to do that. But every design that we made failed to live. So we knew there were pieces missing. And so it took this long iterative process where we had to add sets of genes back to work out which ones were contributing to life. We now have this new minimal cell. It's the smallest genome of any self-replicating species, and it's under scientific review and hopefully will be out soon. But an interesting part of it is over 10% of the genes in this genome are of completely unknown function. I was giving a lecture on a book tour a few years ago up in Seattle, where when my uncle was alive there, he was a lead designer for the 767. And I said, imagine if my uncle trying to design a brand new jetliner didn't know what 10% of the parts did. And he shouted from the back of the room, "What makes you think we knew?" So it's the same process of iterative design and failure, and then going on to the next stage. And so part of the next stage is trying to reorganize the genetic material. So we're using a computer analogy of defragging the genome, where we try to organize the genes by function. 4 billion years of evolution is pretty messy, so we're trying to see if we can clean up some of that mess so that we can actually have cassettes of genes that you can use for design by having them pre-organized by certain functions. And this was a complex process. This is just looking at 1/8 of the genome, all the rearrangements that had to take place to get the genes ordered. So if you look at going from right to left, it's sort of all the changes that took place during evolution. The amazing thing is this works, and we get all the biological functions working totally properly, by having things in a highly organized fashion, which is going to make the next stages of design much more interesting. But the philosophy with all this is to change the DNA software, and you change the species. It was very clear with the synthetic cell, but we're taking it to a different level at Synthetic Genomics, working with United Therapeutics, redesigning the pig genome, trying to insert and replace pig genes with human genes to enable pig organs to be used for human transplantation. So there's about 1 million people that die in the U.S. each year due to the lack of organ transplants being available. People have often thought of using these xenotransplantations, but the organs were rejected very quickly. So it's a very complex process. We started by sequencing the pig genome and then designing which genes to try and change. So we're using a variety of things. We're rewriting sections of the genome. We're using CRISPRs to edit parts of the genome. But we've had some really good successes where for, excuse me, for heart transplants, they're lasting well over a year now. So this is in progress, and just it's amazing changing a few genes associated with the complement system and the immune system change how long these transplants last. But we're optimistic within a few years we'll have a whole new source of organs for transplantation. So we've been putting all these techniques together, tracking as they change, sequencing. Well, there's been very dramatic changes over the last 15 years. There hasn't been a lot of progress on understanding the human genome. And it's in part because we've only had a few genomes to analyze. If they're $100 million each, there's not going to be too many of them done. So we've been watching till sequencing changed to a certain level for us to start up again in this space, and also changes in computing. With distributed computing, with cloud computing, it changes the infrastructure for doing large computes. And also a lot of progress in something called machine learning. Some people in the past have called it AI, but it's much more machine learning of processes. We put all these together and formed the company, Human Longevity. We formed it just about 2 years ago. It's been in operation now for 21 months. We set out immediately to buy some of this new sequencing technology developed by Illumina and PacBio. We have about 50 instruments there and have been scaling up quite dramatically. Each of these boxes cost about $1 million each, but each one of these can do over 1,300 times what the entire setup at Celera Genomics could do 15 years ago. So there we had 350 machines. Each one of these is equivalent to 1,350 times what we could do. So it shows you the progress in that time frame, and it's gone from $100 million down to about $1,500 to sequence a genome. These are the PacBio machines that read single molecules that are much more expensive to run, but give us a different view of DNA. Now, our view from the beginning was not just to sequence DNA, but to interpret the DNA, we need all the other information we can get on individuals. So we do extensive phenotyping, including all kinds of imaging. We measure all the chemicals in the bloodstream. We sequence the microbiome, and we can do both tumor and germline sequencing, putting these things together with other phenotype measurements to create these unique databases and data sets that we can use unique machine learning algorithms to try and sort out. What used to take decades or more and very large pedigrees of rare diseases, we can now solve with a single patient. So this was a case that was brought to us by Children's Hospital. It's actually a very rare disease called Delleman syndrome. There's only about 30 cases of this on the planet. Rare tumors all over the body, and I don't know if you can tell from these MRI images, but massive rearrangement and the structures of the brain. Just looking at the brain images, you wouldn't necessarily think this child would be alive, but he is and has some semi-normal functions, but nobody knew the genetic basis of this. So we did what's called a trio. We sequenced the child's genome and his parents, which allows us to work out which traits came from where. But this graph shows something that showed up very interestingly as soon as we did this. You see these orange bars where they're solid orange. That means that both parents have essentially the identical genome in those parts. That says the parents were highly and very closely related to each other. Turns out they were from a small village in Mexico. They're actually second cousins, not first cousins, but from a village with a lot of inbreeding. So that tells you immediately it's not surprising there's genetic disorders, but it wasn't the sole source. In these areas, we found that there were a lot of genes associated with neuronal development that were mutated that would explain probably what happened with the brain. But one very interesting one, the neurofibromatosis gene, which is one that I actually sequenced when I was here at NIH a very long time ago. And this is one people usually know associated with elephant man's syndrome. But we looked further at it, just to give you a feeling of what we can do. The top band where you see the yellow vertical stripe is the child's genome, and the other two are both parents' genomes. Neither parent had this mutation. It was a spontaneous mutation that occurred only in the child's genome. We all have those. It's somewhere between a couple hundred and a couple thousand. So while we clearly derive our genome from our parents, we all have novel mutations that occurred during that process as well. So this illustrates all those, and now we have, as part of the database, just a very clean, clear diagnostic signature of the genes involved in this disease. Now, cancer is one of the areas where genomics is having its most rapid effect. There was almost no sequencing from tumors as little as 3 years ago, and now it's increasing, but still only about 3% of cancer patients have had this available. But one great case is the AOK case with lung cancer. Pfizer had done a phase 3 clinical trial that had failed, and then a retrospective analysis of it looking to see how many of the lung cancer patients had this translocation in the ALK gene. It turned out about 5% of the lung cancer patients had this translocation. And if you have that translocation, the Pfizer drug has a better than 60% chance of working to decrease your tumors. So they turned around and actually got the drug approved based on having this genetic marker to screen patients first. Now, it's very expensive. It's about $100,000 a year, but with the clear-cut indication that it will work if you have that genetic change, most insurance companies actually cover it, and it's also a blockbuster for Pfizer, and Novartis has also come out with a drug that works in the same space. So this started along with work that Genentech had done with the breast cancer, with the estrogen receptor gene. It started to make people think in a very different view about using genetics as a screening tool for drugs, and now that's expanding tremendously. At HLI, we have two major cancer programs. We have one where we comprehensively sequence the patient's genome, and then we also sequence the tumor genome completely at very high coverage. We also sequence the RNA from the tumor, and then we isolate T cells from the tumor and sequence the T cell repertoire associated with recognizing tumor antigens. In addition, we can look at circulating DNA in the bloodstream, and in collaboration with another group circulating tumor cells. So it gives a very comprehensive view. This was a man in his 50s that got head and neck cancer from being exposed to HPV-16. But just looking at his genome, we found several mutated oncogenes, 5 genes associated with his immune system that were mutated that would indicate he was probably immune suppressed. Those two things alone are not a good thing. If you have a suppressed immune system and increased risk for cancer, it greatly increases the odds. Then from the microbiome, we found he had HPV-16 present, which meant it was more with the combination of the others a matter of time before he developed cancer. He did have a neck tumor that was treated initially and basically went away and then came charging back, and it was unresponsive to anything. So we sequenced the tumor, and we found 25,000 mutations in the tumor. 318 of these occurred where they actually changed the protein coding sequences in that cell. So if you have a good immune system and you get mutations that change your protein coding regions, your immune system then recognizes that as foreign DNA and attacks it and get rid of it. So I suspect we're always having partial tumors recognized and cleared by our systems. If you go spend an hour in the sun, you can develop tens of thousands of mutations in your skin cells. We're always getting radiation and chemical mutations. So we have a good clearing system, but if that clearing system gets interfered with or overwhelmed, then you get cancer. Also in the tumor, we found that the HPV virus had inserted at 4 different sites, disrupting 9 genes, literally causing the cancer in the first place. And from sequencing the immune response, we found that there were a very low immune response against all these new antigens. So it was a very bad situation. We could also do pathway analysis looking for other sites that aren't disrupted but have drugs that get targeted for them. The problem is with cancer, as in this case, these things are discovered very late. They're discovered when symptoms occur. He started with a large growth in the back of his throat, and the late-stage tumor was growing so fast you could actually see it change every day. So even though we came up with a lot of approaches and he was started on an immune trial, he died shortly after entering the trial. But had this come up earlier, the team working with the oncologist actually came up with a cocktail of 3 new drugs that we were certain would kill the tumor cells. Plus, what we can do now is we can take these altered proteins into a custom vaccine program where we can make a vaccine specifically for you against your tumor. So this is just getting going in collaboration with UCSD. We have the first patient that's undergoing this effort now. So we collect data from all these different sources. We have hundreds and hundreds of collaborations of well-characterized patients with different disorders, their phenotyping data, et cetera, and we compile these into subject databases. The phenotyping, we decided most of what was out there was not very adequate, so we set up what we called the health nucleus, or named the health nucleus, initially as a comprehensive phenotyping center, and you can see the list of some of the things that we measure, but I'll walk you through some of these. We also sequenced the microbiome. The very first studies on the sequencing the microbiome were done at the Venter Institute by now its current president, Karen Nelson. And fundamentally, we found we have twice as many bacterial cells in our bodies as we do human cells, and there's increasing association of those with disease. Also, I mentioned earlier the chemicals. Our physiology can make around 2,400 different chemical compounds. So we can actually measure these using mass spectrometry in the blood. And if we do this after somebody's had a meal, we find around 500 proteins in a typical person's bloodstream. Most of them, about 60%, are human chemicals from human metabolism. 30% or so come from the chemicals you absorbed in your diet. and about 10% are bacterial metabolites of some of those food chemicals and human chemicals. We can actually track these and put these things together because we get the pathways from sequencing the genome of the bacteria as well. And so we can come up with new signatures of chemicals that show up in the bloodstream that indicate there's these bacterial pathways that are present. Also, the bacterial pathways should tell us which chemicals should be present. So all this is starting to feed back onto itself. We also have a number of different imaging devices. This is the latest GE3T MRI, and we have unique protocols that only exist at HLI and with GE research, but we can do comprehensive brain imaging, and with some software tools developed at UCSD, we can get really quantitative data. It separates the brain into 20 different regions, and we get very accurate volumes of those regions, and we can compare those to each other, and we can see differences in as little as 3 months. We also do what's called DEXA scanning. This is the only device that is somewhat interventional. It has very low dose radiation, about a third of what I got from flying here from California on an airplane at 42,000 feet. From this, we can get an accurate photo of your skeleton and bone density. We can also measure your muscle density and the fat, and most importantly, visceral versus peripheral fat. And I'll show you another version of this. We can see the quality of the joints, and then we can take you into another room and measure your neurological function just on gait and balance. So this magic floor measures quantitatively the pressure on your heels and toes when you walk. We have people walk normally, and then you have to solve a mathematical problem while you're walking. Some people walk into the wall while they're trying to do that, and others do it differently. But just simply your gait can diagnose diseases such as Parkinson's and autism and a number of others. But it's a clear indication of overall neurological function with these gait and balance things. MRI imaging has changed just in the last year or so with this new thing called diffusion-weighted imaging. MRI measures the motion of water molecules, but by doing this differential weighting, it can find the difference in water molecules in different density tissues. And I'll show you some examples of this. So this is just looking at somebody's carotid arteries. There's no contrast media at all. This is just doing the diffusion-weighted image of what shows up based on the different amounts of water. So if there was any occlusion in the carotids, they would show up very readily. And then we can do the same thing with the brain blood vessels. So some people die from aneurysms exploding. This is a healthy version. So all this data is totally non-invasive of just a part of the MRI imaging. The differential now allows us to see the difference between just a growth and possible cancer. So this is looking at a prostate gland in the normal image, and this is with the diffusion-weighted imaging where there's a tumor. It just shows up very readily. Again, non-invasive, no dyes. And the team we're working with, headed by David Caro, showed that what they get just straight from the imaging matches the best histopathology from actually removing the tumor and doing pathology on it. So we do whole body MRI image, and David and his team can detect a tumor if it's larger than 2 millimeters. But we can get other quantitation for our phenotype measurements, exact density of muscle mass, et cetera. And then there's a unique set of calculations that a team in Sweden developed that converts the same images to give us extremely quantitative on visceral and peripheral fat. And in fact, we can just scan over it and work out exactly how much fat is associated with the liver which is a very key indication of disease. So you can see the green there is the visceral fat, which is definitely, excuse me, the wrong kind of fat to have. I was doing a 30-mile bike ride with one of the fat experts, and halfway through it, he told me that exercise will not get rid of visceral fat. And I said, you couldn't have told me 15 miles ago. So it's only from true calorie reduction can you reduce the visceral fat versus the peripheral fat. So we're getting unique measurements that have not really existed before, so it's getting very quantitative. We also do CT scanning, and this is a picture of my heart from CT scanning. Number one, it proves I have one, for those who have speculated. But you can see the coronary arteries very clearly, but with a calcium score, you can see inside the coronary arteries and get a very good measure on whether they're occluded or not. Again, this is without contrast. You can do it also with contrast. We have the latest 4D ultrasound, and in a very short visit, a little over 30 minutes, we can get a very quantitative view of your heart. So this is part of the video just that each person would see. So you can see all 4 chambers of the heart very clearly, and if the valves are working. And then we can click and then zoom in on any valve and get a very clear picture of it working. But we can also use Doppler shifts, and you can see the directional flow of blood, so you can see if any's leaking back. Now, the old-fashioned way of discovering that is with a stethoscope and hearing a heart murmur, and then physicians spend a lot of time training to do that. In our clinic, you won't find any stethoscopes because it's a very imprecise tool compared to what we measure digitally. Now, when we look at the causes of death for people in the 50 to 74 age range, which includes a fair number of us here, men in that age range, we have about a 30% chance of dying between 50 and 74. A third of that is basically from cancer, and a third is from heart disease. Women only have about a 20% chance of dying during that same time period. 2/3 is cancer and heart disease, but you can see a very different ratio, a higher percentage of cancer and lower incidence of heart disease. So the numbers are quite clear-cut, but even so, we were still surprised when people in their 40s to 70s, just totally people, healthy people, active people with no symptoms whatsoever, about a third of them show up with very complex, potentially life-threatening disease. From the 4D echo, we have 2 individuals that have a significantly dilated aorta. One individual is an OB/GYN doc who has uncontrolled hypertension and lifts heavy weights. So it's like the perfect triangle to put too much pressure on. His first symptom would have been sudden death, and so he's getting that corrected. The stick on MRI, when MRIs first came out and people did whole body scans, they came up with lots of so-called incidental omas where they didn't know what they were, and the claim is people had to get needless surgeries or something to get them characterized. But today it's very different. So this is a patient, an individual in his 50s, totally asymptomatic. He and his wife both in great apparent health, that just came in to get a baseline analysis done. And the image, I don't know if it shows up in that red circle there, but with the differential imaging with water, tumors just light up, as you can see on the right. Again, this is no contrast media or dye. He was about to leave on a European vacation. We convinced him otherwise. This was diagnosed radiologically as a thioma. He went to Miami with a surgeon we recommended and was removed robotically using our MRI image data. He was back at home the next day. They agreed it was a stage 1 to 2 thymoma. It had not started to penetrate into the tissue. If it had, his survival would have been probably less than a year. As it is, there's no follow-up treatment necessary. It was detected early. So this gentleman is part of the as many as 1.1 million new cancer cases a year that when they grow significantly, they cause symptoms. Well, most of those people have their tumors before they cause symptoms, but we don't like to screen healthy people in this country, which makes no sense at all because it's not like those tumors just miraculously appear at the last second before they cause symptoms. So here's one, if he had waited till there were symptoms, there's would have been little treatment and he probably would have survived less than a year. Now his longevity is as good as it ever was if he was tumor-free. They're an interesting couple. This is what we found on his wife, a grapefruit-sized ovarian cyst that's of a size to be clinically significant. We don't know yet whether it's a tumor. In this case, it almost doesn't matter. When you get a mass that large and that heavy, they actually can cause all kinds of damage just by twisting. It can just fall over in the abdomen, choking off lots of blood vessels, and women that have these usually appear in the emergency room in extreme distress. So she's having this removed. We'll find out whether it's a tumor or if it's benign. Either way, it's a very key finding. Here's a man with a brain lesion that's about the size of a large walnut. This is not a tumor, it's just a growth of blood vessels, a meningioma. But this is a person who should not play football, would not be good to be heading balls in soccer. Just a blow to the head could cause that thing to bleed, probably threatening his life. And so the neurosurgeons are recommending that it be removed. You can see it's right at the top of the brain, just a depression there, but he has no symptoms that he's aware of. So another individual, we found a greatly oversized prostate gland that with the differential imaging shows signs of cancer. Again, he was unaware if we had this, and this may be the most benign thing. It's just a large sinus growth that this individual's having checked. But we're well over 30% of the patients coming in finding significant clinical findings just from the imaging alone. Tests looking at genomes also finds the same thing, about 30 to 40% of the population. A study done by Tom Caskey at Baylor College of Medicine looking at YPO members, about a third of them at age of 40 or less had very significant clinical findings just from genome sequencing. So the genomic data, as we get it, is adding to these, and these are just from the last few weeks. We take history and medical records, we go through the imaging, and we try and give this information back in a complicated avatar. So if you come out and go through the health nucleus, you would go to our avatar room where we take a complete 3D photo of you that we can then make a complete accurate avatar of you. People in Hollywood pay large amounts for these just to have them self-inserted into video games. So if any of you are gamers and you want your avatar, your own avatar to play in games, that's possible as well. But we're doing it in part for some of the actual measurements and and trials, as I'll show you in a minute from the face, but also it will be to walk you through your genome and take you through the data. So once these avatars are created, there's software that can have the avatar go through any kind of normal movements. When I'm not there, they have my avatar do a very bizarre dance. I discourage it certainly when I'm around, but we're trying to make it a learning association to teach you about all these different measurements about your genome and how you can learn from it. So it's hard with screenshots when it's, you know, a dynamic setting, but we'll have videos of this in the future. The other thing we're doing is adding remote sensing. Some of you are probably familiar with wearing these so-called Holter devices trying to detect arrhythmias. There's now just a little digital device. It's a little patch that if people can tolerate it, wear up to a week, that records their EKG and events for a week. And it's waterproof, so it can be worn in the shower or any place else. And we have another band that people wear that records all the chemicals they're exposed to for a week. So remote sensing is becoming more and more a part of medicine and digital information. Remote blood pressure sensing is still not up to the standards we're looking for, but people are working hard on this. So we're integrating all this data into a very large database. We have 20,000 genomes with complete phenotype and clinical data now, and this is some of our collaborators. Our goal was to get 1 million by 2020. It looks like we might exceed that by a lot because we're starting to work with insurance companies that have very large numbers of clients that are very interested in this. We're using the machine learning to take all this data and try and make new interpretations from it. And I'll walk you through one of the most exciting ones in terms of phase predictions. But basically, the challenge is converting all this data into knowledge and meaningful information. And we've set up a system that we think can predict anything that can be predicted. You just have to be smart enough to know what to ask it. And that's a challenge in the range of everything we cover. To put it in context, companies like 23andMe use a gene chip that measures common alleles in the population. That would just be that small box on the right. In our database, we have over 60 million variants that only occur once in our 20,000 population. And we saturated all the common variants at around 8,000 people being sequenced. So if we sequence one of your genomes, we would just be adding these rare variants. And just like the ALK translocation, our key medical events, our key drivers of our phenotype are these rare variants. They're not the ones that are common in the population. We can start to predict your age just from your genetic code from a number of parameters. As we age, men lose Y chromosomes. Women lose their X chromosomes. So you can see things start to show up with that. We can measure a number of parameters, including telomere length, that are, with our measurements from doing it from sequencing, getting much more accurate for predicting age. And it turns out changes in your mitochondria. In our mitochondria, we start to lose our factories that generate our energy. We see lots of damage there that we can correlate with age as well. So if you'd asked me 10 years ago, I'd say you get your genome sequenced once unless you have cancer. And you'd probably ideally do it at birth. But now that we can predict your age from the genome and the cost is coming down, it gets very important as we can measure these throughout life. Using some of the 3D imaging, I challenged the team doing machine learning to see if we could predict people's faces and voices straight from the genetic code. And we had a 1,000 volunteers that we paid them about $100 each to take all these tests, have their photos taken, and to donate their genomes to the project. And we start with obvious things like eye color and skin color. We can now predict eye color better than people can self-describe it. Most people have up to 18% difference between their left and right eyes that they're not even aware of. So those predictions are pretty good. And then they brought this green prediction in. It was from my genome, but nobody knew it. And they showed it to my wife and others and asked if it resembled anybody. And my wife immediately said it was either a picture of me when I was younger or a picture of my son. So I had to dig and find some pictures of me when I had hair, so you can see how long ago that was. But now we've added a number of parameters. Here's an earlier picture of me with Ham Smith in the 80s. And so we made this straight computer prediction from the genome and tried to overlay it. It's totally logical that your genome predicts exactly what we look like. Any of you had more than 2 kids, they come out unique at birth, and we can even tell identical twins apart, so it is our software that codes for this. It's to some great extent driven by the predictions of ethnicity. So here's a woman of very complex ethnicity, and here's the prediction. So the top 3 photos are the actual ones from the 3D predictions, and the bottom are straight out of the computer. It's still somewhat of a fuzzy image, as you can see, but as we keep having a larger training set, this gets better. So here's a woman of different ethnicity. The actual photos, if you want beauty photos, don't have them taken with a 3D camera. They're not very kind to people's faces. So we actually smooth them so they'll look more similar to the computer predictions. The other thing the computer predictions right now do is assume that we have symmetrical faces. Now, one measurement of beauty most people have is symmetrical faces, but we all don't have symmetrical faces. So when people see these smooth, totally symmetrical images of themselves, they really like them. So you can see the predictions keep getting better, and we're adding other features off the MRI, predicting skull shape accurately, predicting ears, hair color, hair. Turns out baldness, we still can't predict that very well yet for some reason, but these are pretty stunning when all this is just a DNA sequence file. So the forensic applications of this are obviously tremendous. If you think of what goes on, if you watch CSI from a DNA sample, right now we use statistics to say whether it matches somebody perhaps that committed a rape. And we're limited to people that are in the DNA databases already. With these images, we have at least the whole internet available, so we are using these new image scanners that can scan a photo and then find that person on the internet. So it changes the range of it tremendously. And also, it'd be interesting to see how many here would be interested in. We're from circulating fetal cells where they're used for diagnosis. We can also predict what that couple's child would look like roughly at age 18. We didn't know what genomes would predict, but when you think about it, it's logical. They don't automatically predict your picture as a baby or as a 99-year-old, so it's just post-puberty. But we now have software that can age or de-age photos. Voice is really interesting. It's harder to predict from the genome right now, but we're getting there. We can, just from a digital recording of your voice, we can actually tell whether you're a male or a female. You can see there's a very small overlap, and we can predict your age highly accurately, and now we can predict your height, all from a voice recording. And we're finding now correlations between the voice and shape face, which isn't terribly surprising, but basically everything that's measured gets measured across the genome, and things become a surrogate for some precise measurement. That's all genetics has done basically forever. BRCA1 and BRCA2 tests will not predict whether you'll get breast or ovarian cancer. Basically, the range is 0 to 100%. And about half of women that have both genes positive will not in their lifetimes get either breast or ovarian cancer. But if you have a strong family history where every woman in your family has breast or ovarian cancer, then those tests together with that information become more predictive. So it's a surrogate for something else, and we're used to surrogates. But now we're trying to combine all this data with machine learning that gives us a very different view as we grow these databases. Well, if you want to know more about any of these organizations, here are the websites, including if you're interested in going through the Health Nucleus, you can get information at healthnucleus.com. Thank you very much for your attention.
Would there be any questions, a couple of questions for Dr. Venter?
And we have microphones to circulate so that those watching online can hear you as well.
One back.
If you wait for the mic for a sec, please. There you go.
Once you gather all this information, are you guys working on any therapies alongside?
So we're certainly working with pharma companies that are trying to use data to better predict who should get drugs and who shouldn't. We have our own stem cell therapy group with Bob Harari looking where stem cells can actually help rewrite this accumulation of errors that we see in the genetic code. And we're also doing it, as I mentioned, for cancer-specific vaccines. We're making the data available to others because it's a rich set that covers all human biology, so we hope there will be lots of new therapies that come out of the use. Yeah.
So basically, I do okay. So basically, I do understand that these are like pioneer works, and they're pretty expensive. And just for the future, I wonder if that longevity project will be really directly correlated with the financial status of the person.
Well, you mean what we find is determined by how much money you have? Well, to some extent, social economic status does help determine health status. But cancer is pretty blind to your social economic status. It's pretty much invariant across whether you're a billionaire or live in a trailer. So I don't think it shows that benefit. It's a question of most insurance companies yet do not cover these tests for cancer, even though the outcome can be extremely dramatic. So at the moment, we're dependent on people who can self-pay. But if you read about the program we announced in South Africa, the clients of Discovery Health can get their whole genome, a whole exome sequence for $250. That's about the cost of 1 month of cable television here. And I argue you'll learn a lot more from reading your genome than you will for watching television from us. So I think these will be available for all levels of economic status.
One final question.
Hello. Is it showing? Caduceus is a expert system it's now being used for teaching medical students. it would seem to me that the next generation of it could be developed fairly quickly out of the information you're providing and really enhance the ability to have medical school graduates be able to utilize all of this information fairly rapidly. What would you think?
Yeah, Marty, look, I totally agree. It's the biggest frustration is actually training physicians now. Individuals are very willing. We require them whenever possible to have their personal physician come in or get training to deal with all the data out of the health nucleus. It's about a third that don't even want to. But when you poll, and I always take polls to different groups, when you poll medical school classes now, roughly 2/3 say they'd really like to get their genome sequenced, and a third don't want to. I sort of find that with most groups. There's a third that's really resistant. But if we're constantly training out new physicians every year across the country that themselves are afraid of this information, they're certainly not going to be a good conduit for their patients. I mean, it's a disaster constantly being made where people's lives could be saved by just having this data on their tumors, of knowing which drug to use versus just a blind protocol. So working with David Brenner at UCSD, we're trying to implement it where every student has to, in fact, sequence and analyze their own genome and start to use it in clinical medicine. We'll probably get legal restrictions. It used to be every medical student had to do their own genetic karyotype. And then they kept coming up with so many bizarre versions of genomes just at the gross structural level, and they were getting sued, so they stopped doing that. So, but physician training is the essential part of this. Right now, it's going to be patients training their physicians and introducing it. We have groups of physicians that are just so excited they can't get enough involvement and come to the health nucleus themselves. I'd say good 50% of people coming through are physicians that want to learn about themselves. And usually they have a slightly higher disease category than others do. But it's a challenge. Medical education going forward can't rely on the way it's done in the past. And this machine learning with this data, it's just too much for any one person to know. But we have the ability to use computers to make associations that aren't even obvious to us mortals right now. So.
Well, thank you so very much, Dr. Venter. And what an interesting and thought-provoking and encouraging lecture. Thank you so very much. But now it's my pleasure to introduce Dr. David Penrose, who's the chair of the CSSP Awards Committee and the chair-elect of the CSSP Executive Board. David?
Good evening, everyone. Have to be careful. On behalf of the CSSP, it gives me the greatest privilege to present you the CSSP Award for Leadership and Achievement. This award is given to individuals who merit special recognition for outstanding achievements in the last year in support of scientific research.
Great. Thank you very much.
Be careful how you handle that.
Yes, exactly. Thank you. Well, thank you for this award coming from your Society of Scientific Presidents. Adds its significance, and thank you to the Kavli Foundation for your support of this. This will take a nice place in my trophy case, so thank you very much.
Thank you again. Thank you again. Our next Kavli lecture will be May 9th next year. We are pleased to announce that we have Dr. Jennifer Doudna from the University of California at Berkeley. Dr. Doudna is a leading authority on gene editing. So mark your calendars and look for the advertisements for this special event. And with that, I'd like to thank everyone for coming tonight and have a good night.